Hey there! Today we're diving into the fascinating world of vaccines. These tiny heroes have revolutionized medicine and saved countless lives. Imagine a world without vaccines. Diseases like polio, measles, and smallpox were once widespread, causing fear and suffering. Vaccines have changed all that. They've given us the power to protect ourselves and our communities from deadly diseases. But how do these incredible inventions actually work? Let's take a closer look. Before we can understand vaccines, we need to talk about your immune system. It's a complex network of cells, tissues, and organs that work together to defend your body against harmful invaders. Think of it as your body's own personal army, always on guard against invaders. This army is made up of various types of cells, each with its own unique role in protecting you. These invaders, also known as pathogens, can be viruses, bacteria, or other harmful microbes that try to make you sick. They are constantly trying to breach your body's defenses and cause infections. Your immune system has an amazing arsenal of weapons to fight these invaders. These include physical barriers like your skin, chemical barriers like stomach acid, and specialized cells that can identify and destroy pathogens. White blood cells are the soldiers of this army. They come in different types, each with a specific function, such as identifying invaders, producing antibodies, and directly attacking pathogens. They patrol your body, searching for anything suspicious. These cells are constantly on the lookout for signs of infection or disease, ready to spring into action at a moment's notice. When they encounter a pathogen, they launch a coordinated attack to destroy it. This involves a series of complex steps, including recognizing the invader, signaling other immune cells, and ultimately neutralizing the threat. This intricate process is what keeps you healthy and able to fight off illnesses. So, where do vaccines fit into all of this? Vaccines are like boot camp for your immune system. They introduce a weakened or inactive version of a pathogen to your body. This training exercise allows your immune system to learn how to recognize and fight the real deal without making you sick. Vaccines therefore leverage one important aspect of your immune system: immune memory. Whenever your immune system responds to a pathogen, it activates two types of immune cell: B cells and T cells. B cells are responsible for producing antibodies that neutralize foreign invaders, while T cells are responsible for providing cell-mediated immunity. They recognize and destroy cells infected with a virus. Importantly, B cells and T cells stick around long after the initial threat has been neutralized. This immune memory is reactivated whenever you encounter the same pathogen again. Vaccines stimulate the production of memory B cells and memory T cells. This immune memory induced by the vaccine provides your body with long-lasting protection against the pathogen. Think of it like showing your immune system a wanted poster of a pathogen. This way, if you ever encounter the real thing, your immune system will be ready to fight back. Traditional vaccines have been around for centuries. They work by introducing your body to a weakened or inactivated form of a pathogen. There are two main types of traditional vaccines: live-attenuated vaccines and inactivated vaccines. The earliest vaccines developed for humans used weakened or attenuated viruses to trigger immunity without causing the full disease, and ideally, without any symptoms. For instance, the smallpox vaccine utilized cowpox, a related pox virus that provided protection against smallpox without typically causing severe illness. The rabies virus was the first pathogen to be deliberately attenuated in a laboratory to create a vaccine for human use. This weakened form can still replicate or make copies of itself, but it won't make you sick. Inactivated vaccines, on the other hand, use a killed version of the pathogen. This type of vaccine is produced by inactivating a pathogen, often through the use of heat or chemicals like formaldehyde or formalin. While this process prevents the pathogen from replicating, it remains intact, allowing the immune system to recognize and respond to it. Live-attenuated vaccines use a weakened or attenuated form of the pathogen. The weakened pathogen can still replicate in your body, which mimics a real infection and triggers a strong immune response. Because the pathogen can still replicate, live-attenuated vaccines often provide long-lasting immunity with just 1 or 2 doses. The MMR vaccine, measles, mumps, and rubella, is an example of a live-attenuated vaccine. However, there's a very small risk that the weakened pathogen could mutate and become dangerous again, although this is unlikely. Inactivated vaccines are like showing your immune system a mugshot of the pathogen. The killed pathogen can't replicate, but it still contains important proteins that your immune system can learn to recognize. Inactivated vaccines are generally considered safer than live-attenuated vaccines because there's no risk of the pathogen becoming active again. However, they may not trigger as strong of an immune response, so you might need booster shots to maintain immunity. The influenza vaccine, or flu shot, polio and hepatitis A vaccine are examples of inactivated vaccines. Now, let's talk about a game changer in the world of vaccines: mRNA vaccines. These innovative vaccines have revolutionized the way we think about disease prevention and treatment. Unlike traditional vaccines, which often rely on injecting a weakened or inactivated form of a virus or bacteria to stimulate an immune response, mRNA vaccines use a completely different approach. This new method is not only groundbreaking, but also offers several advantages over conventional vaccines. Instead of injecting a weakened or killed pathogen, mRNA vaccines deliver a message to your cells. This message is carried in the form of messenger RNA. The mRNA acts as a set of instructions, guiding your cells on how to produce a specific protein that is part of the pathogen. The message instructs your cells to make a harmless piece of a pathogen, a protein. mRNA vaccines rely on your cell's own molecular machinery to decode the mRNA and translate that message into a protein. The mRNA from the vaccine is loaded onto specialized cellular structures known as ribosomes. These specialized intracellular machines read the mRNA nucleotide message and translate the mRNA nucleotide sequence into a string of amino acids. The ribosomal machinery then produces a fully formed protein, which is comprised of that specific string of amino acids. The mRNA vaccine often contains the code to produce a spike protein found on the surface of the virus. Once your cells produce this protein, your immune system recognizes it as foreign and begins to mount an immune response against it. This process trains your immune system to recognize and fight the actual pathogen if you are ever exposed to it in the future. The beauty of mRNA vaccines lies in their ability to be developed quickly and their flexibility. Scientists can design mRNA vaccines rapidly in response to emerging infectious diseases. Additionally, mRNA vaccines can be easily modified to address new variants of a virus, making them a powerful tool in the fight against pandemics. This revolutionary approach not only enhances our ability to combat current diseases, but also prepares us for future outbreaks. The development of mRNA vaccines marks a significant milestone in medical science, offering hope for more effective and efficient disease prevention strategies. As we continue to explore the potential of mRNA technology, we can look forward to a future where vaccines are more accessible, adaptable, and capable of protecting us from a wide range of diseases. Truly, mRNA vaccines represent a revolutionary approach in the world of immunization. In this section, we will delve into the key aspects that differentiate mRNA vaccines from traditional vaccines, providing a comprehensive comparison to help you understand their unique characteristics. So, how do mRNA vaccines stack up against traditional vaccines? Let's break it down into several important categories: safety, efficacy, speed of development, and storage requirements. Both types of vaccines are safe and effective, but they have some key differences. Understanding these differences can help us appreciate the advancements in vaccine technology and their implications for public health. Safety. Both types of vaccines undergo rigorous testing to ensure their safety. This includes multiple phases of clinical trials and continuous monitoring even after the vaccines are approved for public use. The rigorous safety protocols are designed to identify any potential side effects and ensure that the benefits of the vaccine far outweigh any risks. mRNA vaccines are considered very safe because they don't contain any live virus. Instead, they use a small piece of the virus's genetic material to instruct cells in the body to produce a protein that triggers an immune response. This method eliminates the risk of causing the disease in the vaccinated individual. Efficacy. Both types of vaccines can provide high levels of protection. The efficacy of a vaccine is measured by its ability to prevent illness in vaccinated individuals compared to those who are unvaccinated. mRNA vaccines have shown very high efficacy rates in preventing COVID-19. In clinical trials, these vaccines have demonstrated efficacy rates of over 90%, which is significantly higher than many traditional vaccines. Speed of development. mRNA vaccines can be developed and produced more quickly than traditional vaccines. This is because the mRNA technology allows for rapid design and synthesis of the vaccine once the genetic sequence of the virus is known. Traditional vaccines, on the other hand, often require growing the virus in a lab, which can be a time-consuming process. Storage. mRNA vaccines often require very cold storage temperatures, which can pose logistical challenges. These vaccines need to be stored at ultra-low temperatures to remain stable, which can be difficult to achieve in some regions, especially in areas without advanced refrigeration infrastructure. Traditional vaccines, in contrast, usually have less stringent storage requirements, making them easier to distribute and store in a wider range of settings. The development of mRNA vaccines has opened up exciting new possibilities for preventing and treating diseases. Scientists are now exploring the potential of mRNA vaccines for a wide range of diseases, including various types of cancer, HIV, and malaria. Imagine a future where we can use mRNA technology to train our immune systems to fight off these devastating diseases. Vaccines are one of the greatest public health achievements in history. They've saved millions of lives and continue to protect us from preventable diseases. But the success of vaccines depends on all of us doing our part. When enough people in a community are vaccinated, it creates something called herd immunity. Herd immunity helps protect those who can't be vaccinated, like newborns, people with weakened immune systems, or those allergic to certain vaccine ingredients. Let's continue to champion vaccines and scientific advancements that improve the health and well-being of people all over the world. Vaccines are a testament to the power of science and our ability to work together to create a healthier future. Thank you for watching this video. Don't forget to like and subscribe to Science Curious.